Thank you. Hi, everybody. So it's an especial pleasure to be here because of the diversity of interests that you all have. And when I began as a student, I was a molecular biologist and a biochemist, and I came into neuroscience from the molecular standpoint, right at the point where molecular biology and genetic engineering became possible in the nervous system. And so coming in now into an age when engineering has so much to tell us about the function of the nervous system, that combination of skills across disciplines for me is so exciting. So go do great things. If you see anything here where you can help me, please let me know, because I would love to hear your input for how we can do more interesting things with the systems that we work in. So as Bilal told you, I am interested in what nicotine does in the brain, and in particular, I'm going to talk today about depression and anxiety. So we think about smoking as a rewarding thing, but in fact, it turns out that smoking, anxiety, and depression are really highly correlated in humans. And for those of you that don't know, and I doubt that there's anyone in this room that does not know what major depressive disorder is, but it is chronic. It's relapsing. And when you think about psychiatric illness, many people write it off as, oh, well, it's behavioral, it's not going to kill you. Unfortunately, for depression, it is a lethal illness, because a certain percentage of people with major depressive disorder will go on to suicide. So here's a disorder where if we have better treatments and better understanding of the circuits that are involved, we can really save lives. And here's the relationship between smoking. It is not simple, and in fact, we need causal algorithms to try to figure out what causes what. So what we know is that people who are depressed are more likely to smoke, but we also know that people who are smokers are more likely to be depressed. And in fact, this has been done in human populations in prospective studies. In one big study at an HMO, they asked kids at 14, are you depressed or do you smoke? And if you were depressed at 14, it predicted whether you smoked at 18. And if you smoked at 14, it predicted whether you smoked at 18. You know what I'm saying. I probably missed one of theirs. But what it says is that the direction of causality is not simple, and that it is likely that one can lead to the other, maybe in sort of an unfortunate positive feedback loop, but also that there are probably shared underlying neurobiological genetic factors that predispose to both. And what we would like to do is to figure out in animal models what the neurobiological underpinnings of this relationship might be, and how we can perhaps then get an understanding about the normal circuitry that govern the role of nicotinic receptors in these behaviors, and also ultimately, maybe we could actually get to treatments. That would be pretty cool. So these are the molecules I'm going to talk about, and I've been really studying this family of receptors now for a couple decades, which is embarrassing, but there's still so much to know. So these are the nicotinic acetylcholine receptors. So when you smoke nicotine or you give nicotine to an animal in an experiment, this is the primary target throughout the brain and body. And there are all of these receptor subunits, and if any of you have had a neurobiology course, you have heard about these, because they have been the prototype for almost every biophysical, biochemical, genetic study of receptors since actually the early 20th century. And the ones that we know most about are these in blue. Those are the muscle nicotinic acetylcholine receptors. They combine into pentamers to transduce all of the signaling between the motor neuron and the nerve. When you think about moving your arm and you move your arm, it's because acetylcholine is released from the motor neuron and transduced in the muscle by this family of nicotinic acetylcholine receptors. And then there's this larger family here that are expressed mostly in neurons, but also in non-neuronal tissues. In fact, if you look at skin, some of these are expressed in skin. We still don't know why. But they fall into two major families. One family that is heteromeric, that means you need one alpha and one beta at least, often even more complex combinations to make a functional receptor. And then this family that can make homomeric receptors, that means 5 alpha 7s, for example, will make a functional channel. And so that's got implications for the binding of acetylcholine, which occurs at the interface between these subunits. The binding of nicotine, of course. How tight is that binding? How reversible? It has implications for which ions can flow through the channel. In general, these channels are all gating positive ion flow, so either sodium, potassium, or calcium. So in general, they're excitatory, but they also desensitize very rapidly. So once they're opened, they're closed for a much longer period of time than they were open, and which has a lot of implications for how that acetylcholine signal actually is transduced through time, and also how nicotine messes up the transduction by acetylcholine. So we think about nicotine as this drug that does stuff, but we should also think about it as a drug that disrupts the stuff that these receptors normally do. And it's important to keep that in mind. And I'm going to go walk back over here because I can't change the slides without doing that. OK, so if we're going to talk about depression and anxiety, we have one major problem, and that is it extremely difficult to deduce the mood of a rodent in a cage. And in fact, you could either think that a rodent in a cage is supremely happy all the time because they have all the food that they want and there are no predators, or you could think that they're extremely depressed all the time because they have no other interesting things like dirty tunnels to explore. Or they could just be grumpy because you're handling them. Or they could be anxious because they expect you to handle them. But we really have no way of knowing by looking at them in the cage. And of course, any of you who do behavior know that in order to study these complex constructs, we have to come up with some kind of assay that has face validity to whatever it is that we're trying to explore, and that actually has some underlying neurobiology that we can study so that we can get at it. And so what we've come at for this anxiety and depression series of behaviors is the idea that what they have in common, and this is not in any way unique to me, that they're maladaptive responses to stress. We have to respond to stressors in our environment to survive. And some of those are physical stressors like lack of food, where it's quite clear how that has a survival advantage. And some of those are social pressures. For example, if you live in a cage with a very dominant mouse who wants all the food, and you have to placate that mouse who's in charge of all the grant funding in order to actually survive. So those maladaptive responses to stress can be operationalized in many different ways. And we also know that there are underlying neurobiological systems, many of them, that respond to stress. That is, that you get an increase in signaling through a specific neurotransmitter or a specific pathway as a result of stressing a rodent or a human. And then maybe if we study those systems, we can circle back around and start to get measures that we can look at behaviorally in the rodent and also ultimately in the human, either by intervening with some pharmacological measure or through imaging a particular system with PET imaging or some other tracer. And that's how we can get these loops where the rodent informs the human, which informs the rodent, and we can maybe make some headway. So I'm going to talk to you about acetylcholine, and that is not an accident. The endogenous neurotransmitter for the nicotinic receptors, as I mentioned, is acetylcholine. It's a neuromodulator. What does that mean? It means that at least in the human brain or in the mammalian brain, it is not the transmitter that actually transduces immediate fast synaptic transmission. Talk to you about the neuromuscular junction. That's fast synaptic transmission. When you want to do something, the neurotransmitter has to be released in a very coordinated way. The signal is very rapid, and it has to be one-to-one. You don't want your muscle firing when there's no acetylcholine, and you want it to stop as soon as the acetylcholine signal finishes. Very easy to understand. So when the nicotinic receptors were first discovered in brain, people figured, well, it must work the same way in the brain, and that is not the case. Instead, what you have is a small number of nuclei. Here is the basal forebrain complex, and here's another complex in the brainstem, which is right around here. And what you see is instead of having point-to-point connections between single brain areas, you have extremely wide connections of neurons in each of these complexes that seem to coordinate, maybe, the effects throughout other brain areas. And in addition, the receptors themselves are not concentrated at the postsynaptic density the way they are in the muscle. When you look at the muscle, there are these beautiful biochemical structures that bind those receptors together, keep them locally just opposed to the sites of release. Instead, the neurons that express these nicotinic receptors look like they've been dunked in egg white and rolled in nicotinic receptors. The receptors are dispersed across axons, across presynaptic terminals, also at these postsynaptic sites. But you don't have this specialization. And in fact, what it looks like is that these receptors are designed to sense volumetric signaling. Not all of them, but at least a proportion of them are there to sense what's happening at a broader time scale, perhaps, maybe fluctuations in overall levels of acetylcholine signaling rather than this point-to-point signaling. And that's important when you think about these complex constructs like anxiety and depression. These aren't single behaviors. These are coordinations of a whole bunch of behaviors that come together to make these crazy constructs. So what we do know from a long time ago, here's a study from 1996, which seems like a long time ago now. I was not only alive then, I was doing science then. And what we see from this experiment is that if you put a microdialysis probe, and microdialysis probe is simply a permeable membrane that is selective for larger ion or larger molecules, and the pore diameter can be selective for the molecule you're interested in. You put it into the brain area of interest, you let the animal do something, or you do something to the animal, and you can see how much extracellular neurotransmitter, for example, is found in that space. So if you put a microdialysis membrane into the dorsal hippocampus and you stress the animal, in this case it's a physical stressor, and this is restraint stress in a rat, you see that the levels of extracellular acetylcholine go up, and they stay up pretty much as long as that stressor is around, and then they come back down on a fairly long time scale. That's an artifact a little bit of how the data are collected. Because this is measured by electrochemical detection, you need enough acetylcholine in order to get a signal. So pools of dialysate are put together. So you can see these are minutes. 60 minutes to 100, that's an hour. It's a lot of time for neuronal signaling. But what you can see is that acetylcholine is certainly induced by stress. So certainly what you might expect is, let's say that this animal could not decrease their acetylcholine levels after this signal. That might be a maladaptive response to stress, and that might be something that could prolong this acute normal response into something we might call depression, or we might call an anxiety disorder. So we can actually mimic this. And we can do that biochemically. So when any neurotransmitter is released, it goes into the extracellular space. It has, any signal that starts has to be terminated in some way. We know that acetylcholine is primarily terminated by biochemical breakdown to choline by acetylcholinesterase, which is an extremely abundant and very high efficiency enzyme. And at the neuromuscular junction, the whole junction is basically riddled with acetylcholinesterase, so that when the neuron releases acetylcholine, the signal is stopped really quickly. And in the brain, this is also true, but because there's clearly diffusion of this neurotransmitter, it can't be that efficient, because otherwise we wouldn't get these signals we could measure by microdialysis. So there is a way to go up and down in acetylcholinesterase signaling. So what we can do is just block acetylcholinesterase activity, prolong the signal, and get more acetylcholine and ask what happens. And in fact, this was done in the 1970s in humans. And we only discovered this after we'd been doing these mouse experiments for some time. But it really shows that everything old is new again sometime. You're going to rediscover literature from the 1950s at some point, and you're going to say, wow, they were really smart, even though they're really, really old. So these guys were psychiatrists, and what they did was they gave an acetylcholinesterase antagonist to humans called physostigmine. And what they saw was that either patients who had never been depressed or patients who had a history of depression showed or reported symptoms of depression just after this pharmacological challenge. Well, it turns out we can do exactly the same thing. Oh, wait, let me actually give you a side note. So unfortunately for these guys, they actually proposed an entire hypothesis about human depression, that if you have too much acetylcholine signaling, you will be more likely to develop depression, whereas if you have too much norepinephrine signaling, you'll be more likely to develop mania. So cycles between these might explain bipolar disorder, and there may be possible targets for medication here. So they had the misfortune of proposing this hypothesis just as a new medication hit the clinics, and it's called Prozac. And those of you who are old enough to remember when Prozac was released, it became probably one of the most popular drugs ever, for one reason mainly, which is it's very safe compared to the classical antidepressants. Classical antidepressants, if you take too much, actually it's toxic, you'll die. And so giving medications that can kill you to people who are potentially suicidal is an actual big risk. But Prozac you can't really overdose on, and so it became the drug most prescribed by psychiatrists for a long period of time. And what that meant was that everything about what depression does was predicated on the fact that, obviously it must be whatever this drug does. And what this drug does is that it reduces breakdown, or actually reuptake, of serotonin. So just after they proposed this acetylcholine norepinephrine hypothesis, the serotonin hypothesis wiped them away, and this lay dormant for many years. And what it shows me is that fashion can actually erase certain interesting observations, and also that very simplistic ideas about how the brain works, one neurotransmitter does it all, is obviously something that we don't want to fall prey to, and that in our era of much more diverse measurement, we actually don't have to fall prey to, because we can be a little bit more agnostic about the particular system that we're interested in and start to look at circuits and interactions between different neurotransmitter systems. So all of this to say, we could now reproduce, it turns out, this exact phenomenon in mice. And so what we did was we gave the same molecule, physostigmine, crosses the blood-brain barrier, and we could measure its ability to block acetylcholinesterase activity. And so here's just a measure of the biochemical effects of the drug. And what we could see is that as we got to the level at which we got effective blockade of acetylcholinesterase activity, we got more immobility in a number of tests of antidepressant efficacy. Now let me go back a step and talk about how we measure depression in these poor rodents. The way that we do it is that we use pharmacological methods to cross-validate the behaviors that we can measure in rodents. So for example, there are all these constructs in rodents about something called behavioral despair. If you give an animal a stressor, if it keeps struggling against that stressor, we call that an adaptive response. It may or may not be. But if it gives up, then we call that behavioral despair. That is not so meaningful unless we can validate that some way with things that we can evaluate in humans. And so what we see is that these models of immobility really respond to antidepressant treatment the same way that humans respond to antidepressant treatment. And I'll give you some caveats. So the models that we use are something called tail suspension, very simple. Mouse is just suspended by its tail. For a while it tries to right itself, and then after a while it gives up. But if you give it an acute injection of almost any antidepressant that works in humans, it will try to right itself for longer. And so you'll see here that the immobility time is sensitive to either things that increase this stress-like behavior or that increase antidepressant-like effects. We have other tests that are almost exactly like this. So forced swim involves an animal swimming around in an inescapable beaker. When it gives up, it just sort of makes the minimal movements to stay with its nose above the water, and we count that as immobility. Again, if you give an antidepressant, you will increase the time it still tries to escape. And it's not simply locomotion, because you can measure that independently. They don't have a lot of great face validity for human depression, because I rarely put myself, for example, in an inescapable pool of water. Although if I did, I would probably want to conserve energy to survive. So adaptive, maladaptive is a little bit difficult to judge there. But there are other tests that perhaps have a little more face validity, and I'll show you some of those as well. Most interesting, I think, and it's become very widely used, is something called social defeat. And I referred to this a little bit. This is a case where you have a dominance hierarchy among mice. You have the big mouse that will beat up, basically, the submissive mouse. And if you expose a submissive mouse to that big mouse for a week, where they're actually not just getting beaten up because that could hurt them, but they get the interaction and then they're separated and they have all the sense, they see that threatening animal right there. Over that week, if you then at the end of the week give them a choice, do you want to interact with an empty cup or another mouse? They will interact with the empty cup. There are some animals that even after that defeat are resilient, and they'll go and interact with a mouse even after spending a week with a bully. And there's actually a lot of work in the field about what is it that makes some of those mice resilient to that social defeat stress, and what is it that makes others susceptible. But again, key here is if you give them an antidepressant, and in this case, chronic antidepressant, which is much closer to what happens in humans. You need at least a few weeks of antidepressant treatment to respond if you're a human. Then you have more time interacting with the other mouse than you do with the empty cup. So again, we use these, in my worldview, as tests of antidepressant efficacy, with the idea that we are finding the same circuits in the mouse that respond to these pharmacological agents or whatever treatments that we have, whether it's stress or something else more adaptive, that we might have in humans. And then we test that. So I try not to call them depression in a mouse. All right, so with that side note, we're talking about now how do those signals from the increased acetylcholine actually result in changes in circuits. So as I told you, there are nicotinic acetylcholine receptors that are widely dispersed along the surface of the neuron cell bodies, axons, terminals. But they're not the only receptors for acetylcholine. There's also metabotropic receptors called muscarinic receptors. And there are two families broadly. There's those that are coupled to excitatory G proteins and those that are coupled to inhibitory G proteins. And they're also found on different compartments of the neurons that are getting input from acetylcholine. So when we tried to induce this state with the increased acetylcholine signaling, we wanted to ask, is this simply the behavioral change that we see some artifact of stimulating the acetylcholine itself, or can we actually find an effect of its signaling through the receptors? Again, we can do this pharmacologically, and so we can give a blocker, a broad blocker of all nicotinic receptors and a broad blocker of muscarinic receptors, or the two together. And we can ask, do we reverse this stress-related behavioral immobility that we saw? So there's that little, here we have an increase in immobility that's blocked by nicotinic receptors, it's blocked by muscarinic receptors, and it's blocked by the two together. But again, this is sort of a broad, it's obvious, right? We increased acetylcholine signaling. We should be able to reverse it with blockers of its receptors. If this is really related in some way to human depression, we should also be able to block it with antidepressants that work in humans. So we used Prozac. And sure enough, Prozac reverses this behavior just as effectively as blockers of the neurotransmitter itself. So here's that increase in immobility that we see in animals given the cholinesterase blocker, and we can reverse that completely with a completely different system. In this case, a blocker of serotonin reuptake, which suggests this is more general in the brain than just increasing one particular circuit that's responsive to acetylcholine. We're engaging circuitry more broadly that can be altered by other neurotransmitter systems. So here we have a system. Now I've shown you that I think it's useful to investigate. Now how do we actually figure out where in the brain this is happening? So there you go. Lots of questions. Fine. So going back to this, we showed that stress induces acetylcholine release, particularly in the dorsal hippocampus. It was a brain area where there was some suggestion that acetylcholine might be intervening. There are other brain areas, but we decided first to test biochemically whether we had some change in the actual breakdown of acetylcholine in this brain area, perhaps as a result of stress. So we did a stress model. In this case, we did the opposite. I got ahead of myself. We did the opposite. We asked if this system is involved in antidepressant-like effects, then we should take antidepressants like Prozac, give it chronically to an animal. We know how they change behavior. Do they actually change the activity of the enzyme that we were interested in, which is cholinesterase? So we simply measured cholinesterase activity in a number of brain areas. And what you can see here is that cholinesterase activity was high in striatum. Prozac didn't change it at all. And instead, the activity of this enzyme was reasonably low in the hippocampus, where that stress-related increase in acetylcholine levels was shown. And actually, fluoxetine increased the levels of the enzyme that we were blocking. So the opposite direction. When we block the levels, we get an immobility response. When we treat with an antidepressant, we actually get more of this enzyme that breaks down acetylcholine. This was just a simple biochemical experiment. It was simply the rationale for us to go in and start to manipulate the hippocampus a little bit more selectively. So one of the things that we can do is that we can use molecular genetic techniques to selectively target particular enzymes, in this case acetylcholinesterase or receptors. Often we do this with nicotinic acetylcholine receptor subtypes. We can do it locally. We can do it with a temporal regulation, because we can put it into the adult hippocampus rather than developing hippocampus. And what we do is that we deliver small hairpin RNAs that target particular coding regions of the RNAs that we're interested in. The dicer complex comes and degrades that particular RNA, and we can get either local breakdown of that RNA, or we can get even cell type selective breakdown of those RNAs now. And what you can see is that we can put these into adeno-associated viruses and deliver them locally. And in this case, the adeno-associated virus also has a green fluorescent protein as a marker to show where we've infected. And we can infect, in this case, the entire hippocampus with a focus on the dorsal hippocampus. And then we can measure in that brain area the actual activity of the acetylcholinesterase after we've broken down the RNA. And you can see we get a significant decrease in activity of that enzyme. And then we can start to measure behavior acutely in the adult. So what we think we should be doing here is that if we break down the enzyme that decreases acetylcholine, we should be increasing acetylcholine levels in the hippocampus. And one of the things that we know is that acetylcholine is signaling throughout the periphery as well as the brain, so there is no reason that this has to be happening in the brain. This could be happening anywhere. But we did think that this might be one place to start. And indeed, what we did was that we showed that when we infuse the small hairpin RNA for the acetylcholinesterase into the hippocampus, we get an increase in anxiety-like behavior. In this case, we're looking at partition between a scary part of a box, which is very light, and a less scary part of the box, which is dark. Or an open side of a plus maze, which is scary, versus a closed part of the plus maze. And what you can see is that when we break down acetylcholine less efficiently, so levels are higher, we have more anxiety-like behavior across these models. Okay. The same is true for all of these behaviors that I described to you as tests of antidepressant efficacy. So what you see here is not only the control experiments that I just showed you, but also another control, which is that these small hairpin RNAs could have off-target effects. So we want to control for them by replacing the cDNA that can be broken down with a human version of the gene for acetylcholinesterase that isn't targeted by the small hairpin RNA. So at the same time we put in the small hairpin RNA, we also put in a replacement for that gene that cannot be affected. And that's this shRNA plus H-human acetylcholinesterase. That's our control throughout all these experiments. So what you can see again is that the small hairpin RNA increases these stress-related behaviors across all of the models I described. So that tail suspension, the forced swim, and the social defeat. And when we put in just a scrambled control, nothing happens. It doesn't rescue that behavior. However, when we put in the human acetylcholinesterase gene, there we get a complete reversal of what we see with the scrambled, small hairpin RNA, right? So what we're saying is, if we destroy acetylcholinesterase, we get more of these depression-like behaviors. If we rescue that with the human version of the gene, which is insensitive to the small hairpin RNA, we don't get those increased behaviors. And here in the social defeat stress, there's just a little bit more of a subtlety. So what we have is two forms of social defeat. We can do suprathreshold, which means that we defeat the animal for a week, and then most of the animals actually will explore that new mouse very little. But we can use a subthreshold amount, so we just give the animals 3 days of defeat. So most of the mice actually don't care. They'll still go and explore the new mouse. What we're looking for when we decrease the levels of cholinesterase is actually more of this stress-related behavior. So we can take these suboptimal defeated animals that don't show an effect on their own, and when we knock down that acetylcholinesterase, all of a sudden now they look like they really don't want to interact with a new mouse at all. And again, we can rescue that with the human construct. So that gives us a brain area. And it suggests that despite the signaling throughout the brain and body, that the hippocampus may be a place to look for effects of acetylcholine signaling on these behaviors. So one of the things that is really important is now to go to the human and ask if this might actually occur in humans as well. So we were very lucky in that we could collaborate with human imagers who are doing PET and SPECT imaging, where they take a ligand that binds to the acetylcholine receptors that is in fact competitive with the endogenous acetylcholine signaling. And I'll show you some data from that. So what do we expect to see? First of all, here's just an example of two individuals that were imaged. The bright colors are where this tracer that binds to the acetylcholine receptors is found in a control non-smoker and someone who is acutely depressed. What you can see is there's a lot less binding of this tracer in the acutely depressed people. We have to do this in non-smokers because nicotine also competes with this tracer. So what you see in a smoker is very, very little binding at all, even less than this one. But when they first saw these images, our human colleagues said, well, so humans who are depressed have fewer receptors for acetylcholine. And we said, well, actually, as biochemists, we can tell you that is not necessarily the case. You could have the same number of receptors, but more competition. You could have the same number of receptors, but you've changed their properties so that they actually have a lower affinity for acetylcholine. There's a lot of things that we could go, or for the tracer. There's a lot of things that could be happening, and we can help you. And here's what we thought that was likely to be happening. If our hypothesis was correct, and these folks with major depressive disorder had more circulating acetylcholine, right? Normally, acetylcholine, which binds at the interface of these nicotinic acetylcholine receptors, would compete with the tracer somewhat in a normal brain. And then you put in the labeled tracer, and it could actually bind to the sites that were not occupied. If a depressed individual really did have more acetylcholine, more of these interfaces would be occupied. Now when you put in the tracer, there should be a lot fewer places to bind, and we should see that lowered signal. So this is simply a survey across a number of cortical regions in patients who are actively depressed, as well as some deeper regions, and in particular you see hippocampus and amygdala. What you can see, which is interesting to me, is that there was no selectivity. There was more competition, or there was less tracer binding throughout the brain. So it didn't look like this was some specific circuit that was misfiring, but instead that there was something more broadly going on. So what we were able to do is to take most post-mortem brains from humans who had died when they were actively depressed, for the most part from suicide, compare them to patients who were controls, who had they were all age-matched and sex-matched, but maybe they had died from a car accident or something that was more acutely traumatic. And also a third group that had a neuropsychiatric disorder, but that was not associated with stress necessarily. And that was brains from schizophrenic individuals. We compared the number of receptors with all of the neurotransmitter washed out in these cortical regions from these post-mortem tissues. And what we saw was there's absolutely no change in number or affinity of these receptors. And so that suggested that this broad change had to be something other than a molecular difference. And we proposed that it might be a difference in occupancy, and then we asked if we could collaborate to test this. So what we did was we asked our colleagues to do a physostigmine challenge, to block the acetylcholinesterase, measure the occupancy of these receptors before and after that cholinesterase blockade, and ask if we saw a difference in occupancy of these receptors in humans. And that's exactly what we saw. So here's 5 individuals before and after acetylcholinesterase blockade, and you can see that in each case the amount of tracer bound was greatly, or was at least measurably decreased. So that gave us the idea that here now we have something in the brain of a human person who is actively depressed that results in more acetylcholine signaling very broadly. And in fact, what's very interesting that I'm not showing you here is that people who had a history of depression were intermediate. So they were not as high as people who were acutely depressed, but now perhaps they were susceptible because they already had more signaling. And we don't know prospectively whether they had higher signaling and therefore they're more likely to get depressed, or whether depression actually results in more signaling over time. And that's an open question that we can ask not only with imaging across time, but perhaps with genetics. maybe there are loci that actually decrease the breakdown or increase the packaging of acetylcholine or increase its release that might in fact be a susceptibility locus for these kinds of disorders. So can we now use this model to ask about the sites and the receptors for the acetylcholine signaling? So we've got acetylcholine increased in human brain. Now maybe the receptors could be a target for therapeutics. So where are they important in the brain? So one thing that we know is that we have very good antagonists. We have great pharmacology for these receptors. They've been around forever. And so we can take antagonists of these nicotinic acetylcholine receptors and ask, if you decrease signaling through them, do you have the opposite effect of increasing acetylcholine? And so that's exactly what you see here. So here you have, again, forced swim test, more immobilities, more stress-like, less immobilities, more antidepressant-like. And if we block all of the nicotinic receptors with this broad antagonist, we get a decrease in immobility. Cool, okay, that's kind of neat. The other thing that you can see here is this other pharmacological agent, which for me is more interesting, because what it is a blocker, a partial agonist of one class of receptors, the one that is most important for reward and smoking-related behaviors, called the high-affinity receptors. But it's an agonist. It increases the activity of a whole bunch of other nicotinic receptors. So the fact that it has the same effect as this broad antagonist starts to give us clues about which receptors might be important to target if we were going to make a drug. And so the question that we can ask at the molecular level is, can we manipulate the receptors that are sensitive to these different agents and ask where in the brain are they involved? All right, so, right, okay. So here is simply another piece of data that this might be relevant to humans. So just as I showed you that Prozac, fluoxetine, could reverse the effects of increasing acetylcholine in the brain, it can act synergistically with these antagonists of acetylcholine signaling to have more of an antidepressant-like effect. So if we use either this partial agonist on its own at a low concentration, we get no effect. If we use Prozac at a low enough concentration, we get not much effect. But if we put the two together, now we get decreased immobility or increased interaction in these two quite different measures of antidepressant efficacy. So we have potential synergy between these different pathways where we can start to ask questions about where in the brain this occurs. So one of the receptors that's thought to be essential for the effects of serotonin signaling is this one subtype. It's called 5-HT1A. It is a metabotropic receptor. It's coupled to G proteins, but it has two different sites of action. One of the sites of action are on the serotonin neurons itself. So 5-HT1A autoreceptors are essential for regulating the release of serotonin throughout the brain. Then there are all of these receptors in other brain areas, including the hippocampus, which you've heard about already. And they seem to be important for transducing some of the signals of Prozac, for example, that lead to antidepressant response. So we can use now targeted knockdown of 5-HT1A to ask which of these different families of receptors are more important, the ones that are on the serotonin neurons themselves or the ones that are in the hippocampus that may also interact with acetylcholine. So here's our strategy. These small hairpin RNAs, as I mentioned, are delivered by adeno-associated viruses. And adeno-associated viruses of this type, which is AAV2, and I think it's not noted here, but AAV2, are actually only able to infect cell bodies. So if you put it into a particular brain area, you're not going to infect the whole brain, anything that targets that brain area. You're only going to affect neurons that have their cell bodies in that area and that make the RNAs locally. Okay? So I can now separate the cell bodies in the brainstem, the dorsal raphe, where the serotonin cell bodies are, from the hippocampus. And what we can do is we can put that either the 5-HT1A shRNA or a scrambled control into the dorsal raphe or into the hippocampus, and we can ask, do we have more or less antidepressant-like effect? And so what you can see is that when we put the scrambled version, so control, into either, in this case, I'm showing you the hippocampus, we can use an agonist of the 5-HT1 receptor, so we can stimulate the receptor that we should be knocking down, and we can get an antidepressant-like effect. or we can use that cholinergic agent and we can get an antidepressant-like effect. When we knock down the receptor, we greatly decrease the antidepressant-like effect of that direct agonist of 5-HT1A. Again, should be obvious. But we also really greatly decrease the effect of the cholinergic receptor agent. And this, again, is a sign that these two systems, the serotonin system and the acetylcholine system, are interacting locally in the hippocampus in order to cause these complex behavioral effects. What I'm not showing you here, because it's all negative, is that if we do any of that in the dorsal raphe, nothing happens. So the animals are perfectly fine. They show a normal antidepressant-like response to either the serotonin agent or the cholinergic agent. So this all sort of points to the hippocampus as a really good place to look for intervention. And this makes sense with human depressed patients because there's been a lot of data on using functional magnetic resonance imaging, fMRI, to show that the processing of stressful stimuli is very different in the hippocampus of patients who are actively depressed and patients who are not depressed. So that makes sense. But is it the case that the nicotinic receptors are also acting in the hippocampus? So we can, again, knock down the receptors. And in this case, actually, first we used an antagonist of one class, the homomeric class of nicotinic receptors, put it directly into the hippocampus, and we got a good antidepressant-like effect. Okay, so locally, the antagonist can work in the hippocampus. This is kind of good, considering that the acetylcholine manipulations occurred there. And we can show that the cells in the hippocampus are stimulated by increasing acetylcholine. So physostigmine, remember, that's the breakdown, blocks the breakdown of acetylcholine. We see more activity. This is a biochemical measure of activity, simply looking at whether an immediate early gene is expressed in those neurons. And that's reversed also by local infusion of this pharmacological agent. But here's the surprising part. Although the activity of those neurons in the hippocampus is decreased by this pharmacological manipulation, the immobility is not greatly changed. So if we knock down this particular subtype of the nicotinic receptor that's the target of that pharmacological agent locally, we get some decrease in immobility, but we don't get a full reversal. And what that told us is that although some of the receptors may be made by those hippocampal neurons, some of those receptors may be coming on terminals from other brain areas. And we wouldn't affect them with this pharmacological manipulation. Now we have to look more broadly in the brain for where these transducers of acetylcholine signals actually are intervening to change this behavior. And we had a number of brain areas that might actually be involved. So we already have some data on the hippocampus. There's a lot of other places that acetylcholine receptors might be involved. So where might we be looking? Well, one place that we looked is another brain area that is involved in stress responses in rodents and also is abnormal in patients with anxiety disorders and depressive disorders. And that's the amygdala, and particularly the basolateral amygdala. So what's the amygdala? It's part of your brain here that's connected widely, again, with other brain areas, including the hippocampus, including the cortex, that in humans has been reported in some case studies that there are people without an amygdala, and those people seem to be extremely resistant to fearful stimuli. They just don't care so much what's happening. In fact, if any of you are climbers, there's a free climber named Alex Honnold who's insane. He doesn't wear any protective gear, and he climbs up like 1,000-foot cliffs in Yosemite. Turns out if you measure his amygdala activity in response to thinking about stressful things, it's very, very quiet. So there's good human data that this part of the brain is involved in responses to stress, your behavioral and emotional responses to stress. And there's more activity in this brain area if you have depression or an anxiety disorder. If you are any of you mouse neurobiologists and you've ever used a mouse brain atlas, you know that you see the pictures of the mouse brain and there's sort of dark colors and light colors. The dark colors are acetylcholinesterase staining. That is the counterstain used for your mouse brain atlas. And the part of the brain that stands out most clearly is this basolateral amygdala. So if you look at the sections through the mouse brain for acetylcholinesterase staining, this part of the amygdala simply screams out. And that's because it's getting a tremendous amount of acetylcholine input from the same basal forebrain complex that I showed you in the first slide is going to the cortex, going to the hippocampus. It's coordinating activity of a lot of brain areas. So it's a good place to look for where signaling downstream of acetylcholine increases may in fact affect behaviors. And what you can see here is that if we look at the overall activity, again, a biochemical measure, not a good electrophysiological measure, but a history of activity of these basolateral amygdala neurons, if we block the nicotinic acetylcholine receptors, we greatly decrease their activity. And it's less clear, and certainly not with these two agents, across other brain areas. So what happens when we go in and we put the nicotinic antagonist locally into the basolateral amygdala? Here, that broad antagonist that blocks all of the nicotinic receptors decreases immobility. In this case, this is the forced swim test, tail suspension test, the same way that peripheral administration of these blockers, it decreases immobility. So again, we're looking at a hotspot for activity that seems to be important for transducing the effects of acetylcholine on these behaviors. So now we can use the same tools, but now we're going to knock down particular subtypes of the receptor. Here's that alpha-7 that I showed you in hippocampus, which had an effect on activity, but not really so much on behavior. Here's another subtype, the main heteromeric subtype. And if we knock down these two cDNAs, we actually get a big decrease in activity, again, measured by total counted cells, so CFOS activity. So that gives us a hint that we are regulating the activity of these neurons by acetylcholine normally, because we haven't stressed these animals. We've simply looked at activity of these cells with or without the receptors that transduce the signal. And then if we look across all of the behaviors basically that I've showed you so far, decreasing the activity of these the number of these receptors can actually alter alter behavior in all of these tests without any stressor at all. So at baseline, when you knock down either this heteromeric type or this homomeric type, you decrease anxiety-like behavior, you get an antidepressant-like response in these immobility tasks, and you get an antidepressant-like response in this social activity response. And we like to see a pattern of behavior across all the tests that's consistent because it goes beyond the individual test, and it comes back to this idea that we have a subset of circuits or a subset of behaviors that are coordinated together. And what you can see is that there is a difference depending on the nicotinic receptor subtype. Whereas you have a consistent effect across all these behaviors with the heteromeric knockdown, the homomeric knockdown is probably best in changing anxiety-like behavior. We do have hints in these tests of antidepressant efficacy. We don't see anything in social defeat, which is this chronic antidepressant-related behavior. So it's not the same. There are subsets of receptors that are transducing aspects of the acetylcholine signal. Maybe this heteromeric type is most important in amygdala at baseline, because there we have an input to the basolateral amygdala that is high, even if you're not stressed, even if you're not undergoing some kind of traumatic experience. So maybe this is important for baseline behavior. If you were lucky enough to be Alex Honnold and born with very low activity in the amygdala, maybe low acetylcholine in the amygdala, maybe you're very resistant to the stressors in your environment that might induce these disorders. If instead you're very sensitive to stress, your hippocampus may be the part of the brain area that is induced as a result of stressful stimuli, and you may have abnormal response to stressors as a result of that component that is not necessarily high at baseline, but only becomes higher when you stress. So it suggests that we do have one system that is innervating these very different brain areas, but that under different environmental circumstances, the different brain areas may be more important. So I'm going to skip ahead a little bit, and I'm going to just show you where we are right now with a very simplistic set of observations that are at the molecular level and the behavioral level for the most part. And this is where we now want to come in and start to ask electrophysiological questions. Can we measure changes in activity in particular subsets of neurons that are consistent with what we've seen so far. So what I've shown you is that if we increase acetylcholine very broadly, we increase a suite of behaviors that are related to stress, whether it's anxiety-like behaviors or depression-like behaviors. And that suggests that this basal forebrain complex, which has the cholinergic neurons that are projecting to all of these brain areas, may in fact be the key node where you might expect either genetic differences to occur or differences in activity to occur in response to stress. So what we're doing now is to label these acetylcholine acetylcholine-making neurons with calcium-sensitive dyes, where we can actually put an optical electrode or look at a slice through that area in response to stress or in response to antidepressants and ask, are we changing subsets of those acetylcholine neurons in their activity in a way that is consistent with the behaviors that we can see? And then we can come in and we can ask, what's driving those neurons? And is their activity coordinated in a way that makes sense with this varied effect of the hippocampus and the amygdala? What we don't know, we know that there are subsets, there's a gradient of acetylcholine neurons across this complex, that some of which go to the hippocampus and others of which go to the amygdala. We don't know if the activity of those different neurons is coordinated, and we don't know if some of them project to both. So those kinds of questions are now being asked, we're asking them in the lab. The other question that we really don't know is how the activity of the amygdala more broadly is controlled by acetylcholine. So what I showed you is that if we block nicotinic receptors locally in the amygdala, and we simply look at a broad signal across all of the neurons of CFOS, it goes way down. So how does that work? Are we decreasing acetylcholine input to every neuron in that structure so that now when we see this decrease in activity, it's the result of just shutting down all of those cells? Or is there a selective cell type specific input? And there have been increasing numbers of electrophysiological studies evaluating the effects of stimulating this pathway either optically, which is very fast, or chemogenetically, which is sort of slow, and looking at the activity of specific subtypes of cells on the other side. And it looks like most of the input of acetylcholine is to GABAergic interneurons. All right, so what does that mean from the standpoint of activity? If the GABAergic neurons are mostly there, and this is not the case, straw man. If the GABAergic neurons are mostly there to silence the principal neurons, and the acetylcholine stimulates the GABA neurons, shouldn't acetylcholine then be quieting that nucleus rather than exciting it? Well, there are a few pieces of information that are interesting. One is that there are networks of GABAergic neurons, some of which disinhibit the principal neurons by having a minus, minus, plus, right? So if you have a GABA neuron to a GABA neuron to a principal neuron, you could excite a GABA neuron and have more activity of the principal neuron. So, okay, so that's easy. We're testing that. We can knock down the nicotinic receptors only in one type of GABA neuron, only in another type of GABA neuron, only in the principal neurons, and ask if that changes behavior and activity. Cool. But more interesting to me is the idea that, in fact, inhibition can lead to synchronization of the principal neurons so that they excite themselves. So if you shut down activity of this network, and that's been shown actually for one subtype of GABA neurons, the chandelier cells, by a group in Australia, when you shut down the whole network, it's like hitting the reset button on your computer. When the activity recovers from that GABA signaling, all of the principal neurons excite each other in a recurrent network, and you get this very large increase in activity that's very prolonged as a result of all of those excitatory neurons firing together. So one thing that GABA signaling, inhibitory signaling, can do is not just quiet the network, but synchronize the network so that now, in fact, you can get much more activity over a much longer period of time than you would normally. I think that's the best way to do it. So I hope that that's what happens. But again, we can test that by a combination of targeting the acetylcholine receptors in only the subtype of neurons that should coordinate this network, and then measuring the activity of the principal neurons using either optical measures or extracellular recording. So I am going to stop there and summarize what I've said. And that is really starting from the human, that there is pretty good evidence that there's disrupted acetylcholine signaling in the brains of actual humans who are struggling with depression. And of course, that's not the only thing that's going wrong, but it is something that's not currently targeted by medications that are prescribed to patients with depression. So maybe it's a way to intervene in a novel pathway that could actually be therapeutically tractable. We know that in general, acetylcholine is thought of as something that enhances cognition. So if you hear about acetylcholine in your neurobiology classes, you probably hear about it as increasing attentional function, or as something that is important for perhaps plasticity that encodes new information. So in my thinking about this system, where we think about not just the acute anxiety that occurs in patients with an anxiety disorder, but the long-term consequences of depression, where you have changes in cognition. One of the things I think too much acetylcholine could be doing is that it is enhancing our ability to encode the bad things that happen. So there's a thing called cognitive bias. So good things and bad things are happening to you all the time. And those of us who are able to encode those good things and actually learn from them and repeat the behaviors that the good things have signaled might be more resilient than those of us who focus on the absolutely horrendous things that really are happening. So my idea in the rodent models is to start to tease that apart. Can I find out if the acetylcholine signaling is really more important for encoding, I got beat up by a bully mouse, than, well, there's a nice mouse over here who'll snuggle with me. If you can actually distinguish those things behaviorally, we can start to tease those apart cognitively in humans as well. So one of the things that we think is that we might actually be contributing to negative bias. That's something that could be theoretically tested and then tractable to an experimental design. And then finally, this idea that there's phasic signaling, that means transient increases in acetylcholine in the hippocampus, that are supposed to be linked acutely to negative events, and that might actually be prolonged beyond those negative events if the brain can't clear that acetylcholine or stop those acetylcholine neurons from firing, that might in fact be very important for this maladaptive effect, whereas the tonic signaling that goes into the basolateral amygdala that clearly gets a lot of acetylcholine no matter what, might make you more or less susceptible to these kinds of stressful life events. And I'm going to stop there because I'd like to leave some time for questions. And I was going to talk about another project on aggression, which is also really interesting and is a literature discovered by a resident, an MD PhD fellow in my lab, who used his time overnight in the emergency ward to start reading papers from the 1960s, which were really interesting. But that will have to be for another time. So thank you very much for your attention, and I will skip forward to show my group. Actually, I want to acknowledge their work, because this is not me. This is their work. And there they are. And oops, go away. You. All right. Or you? Yes. So I particularly want to, there they are, aren't they nice? I want to acknowledge Jan Mineur, who is a long-term research scientist in my group, who has really been carrying out a lot of the work on the acetylcholinesterase project, but also Barbara Calderon, who was one of my first postdocs, who made the first observation that changing acetylcholine levels in the brain could actually change these behaviors that were sensitive to antidepressants. And she discovered some of that literature from the 1970s that suggested this happens in humans too. Reba, when she was a graduate student in the lab, started the molecular genetic experiments to actually target the nicotinic receptors and look at their effects. And then Steve Pittenger, who's a current postdoc in the lab, is doing some of the cell type selective work and in the amygdala, and Rick Krause is doing some of the work on activity in the hippocampus. Lots of human collaborators, particularly Zubin Bagogar, who did that first imaging study in humans, and then Irina Esterlis, who was willing to give physostigmine to human subjects and look at the occupancy of the receptors, and of course our funders. And you for your attention. Thank you. So I'm happy to answer any questions if you have any attention left and your acetylcholine system is still functioning. Super question. We looked at that, and yes, indeed. So if we look, and we've only looked at it one way. So the question is, do we see any difference in adult neurogenesis or the volume of the hippocampus as a result of any of these manipulations? So what's known is that classical or antidepressants used in humans can increase basically both the production and survival of new neurons in the hippocampus in animal models. And we see that with these nicotinic antagonists, but we also see it with Prozac. And what we've done is only one experiment where we took complete nulls for the heteromeric nicotinic receptors, and we asked, in those complete nulls, can we still see the behavioral effects of Prozac? No. And can we see the effects on neurogenesis? And the answer is no. So in the absence of the nicotinic receptors, again, and this is constitutive, so it's not the perfect experiment, we no longer get this change in increased survival of new neurons in the hippocampus. So it's consistent with the idea that they participate in the antidepressant response, and it's consistent with this interaction between the systems. Then we did one more experiment, which is we looked at the volume of the hippocampus in aging without these receptors. And sure enough, the volume decreases with aging much more rapidly in animals without these receptors than with these receptors. We took that to mean that maybe it was a loss of an endogenous neuroprotective effect, but it's likely also that some of these effects on adult neurogenesis may contribute. So they're together. Mechanisms for that still not quite clear. Yeah?
Super interesting talk. I have a question, though, as far as how you're thinking about when acetylcholine is acting to produce these depression effects. Is it because you're giving it chronically, this acetylcholinesterase?
No, actually, we're giving, so the challenge, we have both. So the pharmacological challenge is very acute. It's right before the behavior. But the genetic challenges are completely, are yeah. Yeah.
Where you knock down the shRNA,
yeah.
and then you do recovery. And
Oh, that's right. Great question. Super duper question. So the way that we did the rescue was that we infused the rescue plasmid at exactly the same time as the knockdown plasmid. So they never got knocked down. Now here's the secret. If we give the rescue plasmid 6 weeks later, it no longer rescues.
Do you think of it in terms of compensation?
So I think of it as the kind of plasticity that probably occurs in the stressed brain. So acutely, we clearly have an effect of acetylcholine for mediating those circuits that result in the behavior, and that's kind of consistent with these strange mouse models where an acute injection of an antidepressant changes behavior. The way I think of that is that you have systems that mediate those behavioral changes directly, and then other systems that are involved in the plasticity. So when we look at the inability of acetylcholine manipulations to reverse the phenotype, that suggests we actually have a model where we can look at what those plasticity events are. And I would suspect that those might be effects on adult neurogenesis. They might be effects on the ability of other neuromodulatory systems, including serotonin and norepinephrine, to alter the activity of those circuits. And so I think the acetylcholine is more acutely involved. And there is one experiment in humans that supports that and another that does not. So there have been some small trials of antagonists of acetylcholine receptors that have been done in human depressed patients. And particularly with muscarinic antagonists, they seem to work acutely. So in the case of the muscarinic signaling, it's probably involved directly in whatever those behavioral things are that we call depression. Nicotinic agents, not so much. There you need to give them for 3 to 6 weeks in order to see an antidepressant response in humans.
So it's more setting the baseline.
Seems to be. So again, amygdala, hippocampus, kind of cool. Maybe that's what we're seeing breaking down. Don't know. Okay. Okay. Thank you. you.